Hello friends, in this video we are going to discuss about recombinant DNA technology, in short RDT. So what is recombinant DNA technology or RDT? It is a method of combining DNA from different sources or species to create new genetic sequences or to produce new genetic combinations, either to make copies of that gene or to produce product encoded by that gene. It is also known as genetic engineering, molecular biology technology or cloning. These are of value to science, medicine, agriculture and industry. Now what is recombinant DNA? A piece of DNA that has been created by combination of at least two strands is known as recombinant DNA, rDNA. They are DNA molecules formed by laboratory methods of genetic recombination to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in genome. Now let us see its history. Recombinant DNA in a living organism was first achieved by Herbert Boyer in 1973 of the University of California at San Francisco and Stanley Cohen at Stanford University. They use E. coli restriction enzymes to insert foreign DNA into plasmids. Now let us see the steps involved in recombinant DNA technology with the help of this flowchart. Isolating the target or desired DNA and isolation of vector. Then cutting both with restriction enzyme. Both the target DNA and vector is cut with the help of restriction enzyme. Then ligation and production of hybrid DNA or recombinant DNA, that is known as chimeric DNA. So both the isolated DNA and vector are ligated to produce recombinant DNA, also known as hybrid DNA or chimeric DNA. Then introduction into host cell called transformation. So this recombinant DNA is then introduced into host cell. After that, isolation of transformed cells with recombinant DNA. And transformed cells with recombinant DNA are then amplified, known as DNA cloning. There are two methods of amplification, cell-based and cell-free DNA cloning. Cell-free DNA cloning is done by polymerase chain reaction, that is PCR, which we will discuss in separate video. Now let us understand this recombinant DNA technology with the help of this diagram. This is the foreign DNA or target DNA or the desired DNA. This is the vector DNA molecule. Then it is cut into fragments with the help of restriction enzyme. So these are the fragments of the target DNA and this is the fragment of vector DNA. Then the desired gene is ligated with the fragment of vector by the ligase enzyme, and this process is known as ligation, and this results in recombinant DNA. This is the target gene and this is the vector DNA. It is also known as the hybrid DNA or chimeric DNA. Then this recombinant DNA is introduced into host cell by the process of transformation. So this is bacterium with the recombinant DNA. Then multiplication of recombinant DNA molecule takes place and this gives several copies of recombinant DNA molecule. Bacterial cell also undergoes cell division, and with numerous cell divisions, it results in clones of bacteria. So these are the bacterial colonies growing on solid medium. Now let us discuss the steps involved in recombinant DNA technology in detail. First is isolation of genetic material. It is the first step in recombinant DNA technology. Desired DNA is isolated in its pure form, that is DNA which is free from other macromolecules like carbohydrate, protein, RNA, etc. DNA is separated and purified from other macromolecules with the help of enzymes such as lysozymes, cellulase, chitinase, ribonucleases, proteases, etc. These digest other macromolecules except DNA. Then ethanol is added that causes the DNA to precipitate out as fine thread. Then this is pulled out to give purified DNA. So from where gene of interest is obtained? It is obtained from genomic library, cDNA library, chemical synthesis of gene if the sequence is known. If the number of copies of desired gene is not enough for gene cloning, gene amplification techniques are opted like PCR, polymerase chain reaction. Then second is isolation of vector. What is a vector? Any DNA molecule that has ability to replicate inside the host to which the desired gene has integrated. Vectors include plasmids, bacteriophages, cosmids, BAC, yeast vectors, shuttle vectors, etc. These are circular DNA molecules. Now third is restriction enzyme digestion. Restriction enzymes act as molecular scissors that recognize and cut DNA at specific sequences, which often create sticky ends for easier joining. These reactions are called restriction enzyme digestions. Let us see the process of restriction enzyme digestion. Purified DNA is incubated with selected restriction enzyme at optimal conditions for that specific enzyme. Then agarose gel electrophoresis is done, and this reveals the progress of restriction enzyme digestion. DNA runs on an agarose gel and then it is separated out based on size. This allows cutting out and separation of digested DNA fragments. Vector DNA is also processed with same procedure. Now fourth is ligation of DNA molecules, the process of joining two pieces together using the enzyme DNA ligases ligation. These two pieces include purified DNA plus vector, which are cut with the same restriction enzyme. What are ligases? Ligases are molecular glue joining DNA fragments by forming phosphodiester bond. So these join the two DNA fragments, that is purified DNA or gene of interest and the vector, and these are joined by forming the phosphodiester bond between them. DNA molecule resulting from ligation is a hybrid of two DNA molecule, the molecule or the gene of interest and vector. This intermixing of DNA strands is called recombination. Hence, this new hybrid DNA molecule is also called a recombinant DNA or chimera, and the technology by which the hybrid DNA is formed is referred to as recombinant DNA technology. Recombinant DNA are hybrid DNA molecules consisting of autonomously replicating DNA segment plus inserted elements. Then fifth is introduction of recombinant DNA into suitable host. Recombinant DNA is introduced into a recipient host cell. This process is called transformation. In these cells, the chosen replicon can undergo DNA replication independently of the host cell chromosome. Bacterial cells do not accept foreign DNA easily, hence are treated to make them competent to accept new DNA. The process used may be thermal shock, Ca2 plus ion treatment or electroporation, etc. The next is isolation of recombinant cells. Transformation generates transformed and non-transformed bacterial cells. Transformed cells need to be selected from rest of the cells or colonies. So we can say that it involves filtering the transformed and non-transformed host cells. For this, marker gene of plasmid vector is employed. Example of marker gene is antibiotic resistant gene, etc. Now what are marker genes? Genes often conferring antibiotic resistance that are included on vectors to identify and select only those host cells that have successfully taken up recombinant DNA. These are also known as selective markers. Now, the last step is selective propagation of cell clones. It involves two stages. Initially, the transformed cells are plated out by spreading on an agar surface in order to encourage the growth of well-separated cell colonies, called cell clones. In the second stage, subsequently, individual colonies can be picked from the plate and the cells can be further expanded in liquid culture. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.